Okay, great. I think we can get started, but welcome everybody. Hello. Welcome to today's webinar event, which is hosted by the World Neurology Foundation and the AEN Global Health Section members. My name is Andrew Huang. I'm the program director for the World Neurology Foundation, and it's really a pleasure to have you all here to hear from our invited speakers. So I'd just like to start off really quickly to give a quick introduction about the World Neurology Foundation, and before I hand it off to Deanna, who will introduce our renowned speakers for today. But just to quickly introduce, what the World Neurology Foundation is about, we're an international nonprofit organization that has the mission and vision to promote neurology services and education in under-resourced regions around the world. And we had started this webinar series late last year to really help promote the numerous kind of global neurology efforts that are happening and being conducted by researchers, clinicians, and trainees all around the world. So today, we have two amazing speakers who will be talking about their efforts in Africa with stroke genomics and neurobiobanking. And I'd like to hand it off to Deanna actually to introduce our speakers for today.
Thanks, Andrew, and thanks again to everyone for signing on. We are really, really excited for our speakers today. It's my pleasure to introduce Prof. Steve Sarfo from Kwame and Nkrumah University of Science and Technology in Ghana, and Prof. Rufus Akinyemi from the College of Medicine at the University of Ibadan in Nigeria. Both Prof. Sarfo and Akinyemi are renowned clinicians and researchers and have really contributed a vast amount to our understanding of stroke in West Africa and in Africa in general. And so I think it's a great honor to have you both here, and we're really excited to hear about your experience with genetic testing in the SIREN study. So without taking up any more of your time, I will turn it over to the two of you. Thank you so much for being here with us today.
All right, thank you. Rufus, go ahead. Hello, Rufus. Hello. Where is Rufus?
I think there might be like a technical issue. Maybe, hold on.
Has it dropped off? No, he's still on. All right, see you.
Yeah, I think his not screen, his camera, his video is frozen. So let me just quickly message him. Oh, he just dropped off, I think.
Okay. So should I start the... Okay.
Maybe while we're waiting for him to sign back on, do you want to give people an overview of the SIREN study? And then we can turn it over to him when he comes back.
Yes, please. Certainly. Yeah, so thank you very much for this invitation to be part of this webinar. The SIREN study is the largest epidemiological study on stroke in the African continent. And the focus of SIREN was to, first of all, characterize the traditional risk factors for stroke. We had known about the risk factors for stroke, but we didn't have data on the effect sizes of the various risk factors for stroke when we started the study way back in 2017. And I think we are privileged to have Maiwa, who was also a PI for the study, among our attendees. So the SIREN study was conducted across 16 sites in Ghana and Nigeria. We enrolled a little over 3,500 stroke patients who were all characterized using CT scans or MRI. And it included ischemic strokes and hemorrhagic strokes. And then we recruited age and sex match controls. And for the first time, we were able to characterize the various risk factors for stroke. Of course, hypertension was the dominant risk factor for stroke. And we are currently at a point where we are trying to understand the contributions of untreated hypertension, treated but uncontrolled hypertension, and then we're also looking at even those who are controlled with hypertension, what's the effect size on stroke occurrence? So we've done that, and we're also looking at the genetic risk factors for stroke, and that's what we want to talk about this afternoon. Oh, good. Rufus is back. Are you back for real, Rufus, my brother?
Yes, my sincere apologies, please. Due to internet.
Yes, I had to cover by talking about Sarin.
Yes, absolutely.
Yes, absolutely. Of the moment. So
yes, absolutely. You've done a great job. So I just continued from where you stopped. I mean, yes, thanks, Fred. I heard you talking about Sarin material. Yes, not of what we've done so far. So let me just cap that off by mentioning essentially that, well, stroke is a global problem. 1 in 4 persons stroke. From this paper from the GBD study published in the Journal of Medicine. And in Africa, stroke is a very big problem today, but it didn't seem to have been solved about less than 100 years ago. This is one of the earliest papers from the continent to report about hypertension and stroke. And you could see from this paper that was published in South African Medical Journal that native South, Central, and West Africa, hypertension and stroke were rare at the time. About 70 but as we know that Africa today is in a very transition, facing a double burden of both communicable and non-communicable diseases. I mean, we're very familiar with this medical bill, which summarizes what we know about stroke in Africa 15. Then we further updated what we know about stroke, about less than a decade after, essentially documenting the progress that we have made regarding stroke in Africa, stroke research in Africa in the last, it was also a very deep overview regarding stroke across the continent. And of course, looking at what we have at the moment, the progress, and then most of the future priorities that we mentioned, some potential stroke service in development, capacity building, research, and advocacy. I'm sure that many of us are familiar with this review. Now, yes, the Sarin project is essentially looking at genetic and inflammatory status for stroke in Africans. And one of us are familiar with some of the elegant output on the status for stroke. The question is, why is stroke genomics important? Why is it important for us to explore the genomic risk factors for stroke? Well, it's important to provide the solutions to stroke because this will lead to better definition of causal pathways. As we all know, that at the level of genomics, stroke is a very heterogeneous disease in the sense that different genetic variants have been associated with different subtypes of stroke. Ischemic subtypes, other subtypes. Because ICH, when you look at people's genetic analysis of genomics in these areas, you will notice that associated genetic variants are different. Now, so it's also important for us to identify novel therapeutic targets, which is the hallmark of precision medicine, specific targets that it's, that it's just on target with less we feel side effect profile. Of course, to reduce the stroke, it's very important for us to discover new therapeutic approaches, especially approaches that will be targeted to different people, ancestries. Of course, very important so that African and other underserved regions of the world benefit from precision medicine. Now, stroke genomics is so important that we have a global consortium that focuses on this effort. The Genomics and Neurobiobanking consortium. And within this consortium, we had different groups aspects of stroke across different pathways and across the lifespan. And then we are happy to also say that Africa is very well represented within the IHTC.
Sorry, Prof, I apologize for interrupting. We're having a bit of difficulty hearing you.
Oh, really?
Yeah, I'm not sure if you can get a bit closer to the microphone.
Hello, is it better now?
Yes, that's much better. Thank you.
All right. Okay. Right. Okay. All right. Right. Thank you very much. So Africa is also now part of the ISGC, and this certainly will drive forward. We're hoping that in the future, many more African countries will be represented within the ISGC. Now, what are the peculiarities of stroke in people of African ancestry?
I think there's an issue with
We compare data on stroke from Africa, and of course, there's a significantly higher proportion of this kind of hemorrhagic stroke in people of African ancestry compared to other research populations. And more importantly, we seem to have the highest proportion of small vessel disease from all the data, maybe comparable to what you find in Asians, for instance. It was quite higher compared to patient data. Of course, from the SLEDGE study from London, we also understand that genetic heritability in African ancestry is higher compared to European ancestry. And of course, outcomes are comparatively worse. Mortality, mobility, motor disability, and other outcome measures. Yes, even though the worst outcomes are largely due to social determinants of health, systemic factors, but we think that it is not impossible that there may also be something about either the genes or the genetics that do contribute to the worst outcome in people of African ancestry. Now, why is it important to have people of African ancestry get them involved in stroke genetics? We as well know that globally, it's been said that less than 5% of all consumers data sets have contributions from people of African ancestry, especially from Africa. So there's a global move, to improve representation of underserved populations in the spirit of diversity, inclusion, and equity, to increase representation of people of African ancestry in global genomic research. So that is one major reason. Of course, the racial disparities in stroke community, as I mentioned earlier on, also there may be subtle contributions from genetic factors to this disparity. And of course, it's also that because of the unique features, the African genome in terms of the higher heterogeneity, short appetite blocks, and low lipid disequilibrium, that having African data in global genomic efforts actually help us to have better fine mapping and ancestry meta-analysis so that causal variants can be detected with adequate accuracy for these type of implications for downstream mechanistic processes, and for new discoveries that can help stem global type of stroke. Now, this slide, just a general slide, that shows diseases such as stroke. Generally, this applies only to stroke, but most diseases. You may do family linkage studies. This actually started with string studies, varies in the wood past, family linkage studies, and now we also do genes association studies, whereby we look for specific genetic variants, specific single people that are found in one population, examine whether they are present in that population. And then this third approach is genome adaptation study. You can also have whole genome studies, exome studies. The important thing is that as you move from A to B to C, you need a higher, greater sample size for you to be sufficiently powered, to make discoveries. The major difference between B and C is that in B, we are going for specific candidate genes, so it is not no new discovery. But for C and higher levels of which is, it's microscopic approach. We are making new discoveries, and with this discover new variants, it is because you to replicate what your findings, primarily in different modes of select competition. You can also meta-analyze your data with other data sets, and that is the global trend. And then when you find discoveries, then you want to look at the functional implications of the variants that you discover, and then you do a lot of functional analysis. Some of them involve in silico analysis, and some involve functional studies in the laboratory using different animal models, so that we can develop, either you can test an existing hypothesis or new hypothesis, all of which may ultimately lead to discovering new strategies for new drugs. But the ultimate goal always is to intervene to stem the tide of disease in human populations. Now, what do we know about genomics data? From candidate gene studies that have been done on the continent since about almost 20 years ago, about 2 decades now. Earlier studies were Northern Africa, Tunisia, Morocco, and Egypt. And most of these, again, were largely candidate gene studies looking at different genetic variants, blood clotting mechanism, protein E, platelet alloantigen, gene. So different variants of different genes have been studied. That would probably be to this. Then more recently, when Sub-Saharan Africa is now been involved, medical studies from Zambia, led by Dr. variants on protein E in a very small, modest sample size. And then more recently from Nigeria and Ghana, courtesy of the sharing projects. Now, so again, many thanks to the NIH, US NIH, and Wellcome Trust that have enabled to be involved in the genomic evolution in Africa through the sharing projects, especially as a case for study with 3 different thematic areas. Genomics aim was to characterize stroke accurately in Africa, and I think we have a shared program that makes genomics, stroke risk factors, stroke subtypes, and of course outcomes, brain mortality, determinants of outcomes. Genomics, with the response to genomics, it is still work in progress, and of course through this project, bioinformatic capacity has also been built among participants. Signed the silos for projects. So we've conducted some candidate studies, and of course, we've also conducted the first GWAS of stroke in Africans. So the summary of the candidate study, this was a very, very small study involving a little over 1,000 subjects in total. What we did was to examine, to pick out all the different candidate genes that have been associated with stroke from different populations, African Americans, Europeans, and Asians. And we look for them in the African side. Found some variants on some genes, the interleukin 6, the RS 18796, and the second dependent inhibitor gene were found to be associated with stroke among the genes, as well as variants in the well-known gene, the HDAC C9 polymorphisms was found to be associated with small vessel on becomes degenerative becomes participate in sharing study. But very interesting that the first edition was Second was across both genders. The interesting thing about the April one was that shortly after the April one paper was published by our team, the regards team in the US, confirmed that that association by publishing, a similar finding in the American subcohorts of the regards study. Now, the interesting thing about the April one thing is that it's associated with chronic kidney disease, full of impact ancestry. And then when you look at the architecture of the brain and the kidney, small vessels are involved in both organs. And so it appears that there's something about small vessel conditions and the World War gene. So whatever defines small vessels, the disease, and may have some role to play. Of course, we are still trying to explore this in the bigger studies, GWAS and other approaches that we are using. Now, for GWAS, and not previously suspected diseases of interest. So for the GWAS, we used the East Africa array chip production, 5 million sleep array, used the Illumina with rich content from people of African descent, also including variants systems treatment that make disorders. So we genotyped 1,743 stroke cases and 1,612 stroke-free controls, all recruited from 15 sites in Nigeria, Ghana. Of course, we recruited data sets into 3 different reference panels, the each technical one, the system of genome research and the complement, and multiple control steps on take. The slide shows the table comparing the stroke cases and controls, and we can see that there's significant differences between the case and control with respect to age, cardiometabolic factors. We then developed models for the association analysis using logistic regression models, using about 8 different models, including all the low steps for analysis of data. And then the principal components analysis did show, you could see the distribution of individuals from the silent cohorts is similar to the distribution that was reported in the 1,000 genome studies. And this was a test, to show kind of validity of the distribution of the genetic variant. What are similar to what have been previously discussed. So adjusting for different covariates in the model. And then I think the plots, is close to normal. This allocates that zooming 3 and 5 respectively, where we think our most significant sleep were located near the MIF gene, 3, near the 813 population 5.
Rufus, will it help?
Yes, sir.
If you put off your camera?
Okay, all right.
Let's try that and see. Thanks.
All right, okay. Right. So we made effort to, because of the absence of an existing suitable African cohort for replication, the best we could do was a sort of transferability analysis in African American cohort in the COMPASS study, which is an older study that has gathered together minority populations, including African Americans from North America. And the mega stroke is a bigger study that included populations from Europe and North America. So we made effort to, to replicate what we found the side cohort in these two different cohorts. What you could see on this slide are some of the variants in the compass in terms of allele frequency was similar to those desiring in terms of allele frequencies of the different variants that were detected in the compass and then compared to the siren. Oh, hello, can you hear me, please?
Yes, it's a bit on and off, but we can hear you now.
All right, okay. So, but if you look at the mega stroke cohort, the distribution of the mega stroke and the compass with respect to siren, you could see that the distribution is, there's no linearity to the distribution, and that shows that the allele distribution of the different variants differ between the megastroke and megastroke siren, just showing that the megastroke population is a very different population from other the indigenous population or the African American population US. On more granular details, a few SNPs show some nominal significance in terms of replication in the African American cohort. We also did some association analysis looking for genes on chromosome 5, 3, and 5 that were in association with the variants interests. This showed significance in the siren cohort. And the interesting thing about this is that many of these genes, these show important in terms of functions. Many of them, in terms of expression, were expressed either in vascular tissue, brain, or some even associated with, with cerebral vascular disorders. So, in summary, we then summarize that the major strength of these studies that is the first ever study was in the African population, and then the loci found to be significant, actually regulatory in the pathology of ischemic stroke, including those variants that were in association with the key variants of interest. Because the major evidence that was a stable replication cohorts was not available. And then sample size limitation regarding subtype analysis. Of course, we can perform meta analysis and transcendency meta analysis in cohort, but none of them demonstrated genomic significant replication in the decided setting. Of course, there are also all the transitional genomic approaches, in other words, to ensure that we carry our community along. One thing we found it is very important subjects. Post-case control was that there was high acceptability and willingness to undergo genetic testing. This has implication for downstream, willingness of the people to undergo genetic testing if it becomes like a kind of care testing that you can do. Other aspect of stroke control have been studied, attitudes, behaviors of the populace, this process ensure that so I will hang up the story about stroke genomics in Africa where we are at this point, and then hand over to my brother French, who will speak. Very much. So back to you, Fred.
Okay, thank you very much, Rufus. Let me see if I can share slides. Share. All right. Can you please see my slides?
Yes, we can, but maybe you can put them in presentation mode.
Yes, please.
Perfect, thank you.
All right, so I'll switch gears and we'll start looking at the issue of neurobiobanking in Africa. Most of the data or most of the samples that we require to be able to analyze and come out with data on genomics of stroke come from our participants who generously contribute their samples to this analysis. And so we started a study called the ELSI study. And the aim of the ELSI study was to identify, examine, and then develop novel interventions for addressing the ethical, legal, and social issues regarding stroke genomics and biobanking within the African context. The aims of this study, the ELSI study, was one, to look at understanding from our participants in Ghana and Nigeria, their views on donating blood samples for genomic research, storage and reuse of, stored blood fractions, and return of individual research results and incidental findings back to them, and as well as, understanding biorights, governance, as well as the legal and ethical issues of, how do I get this up? Let me see, sorry. Yes. So whilst doing this, we seek to develop novel interventions, because what we are realizing is that most of our participants really don't understand what even giving your informed consent to have your samples taken and analyzed for genomic research all means. So we seek to design interventions to improve that understanding among our participants. So we believe that neurobiobanking is important in the African context because it provides opportunity for deeper learning. And we say that neurobiobanking just simply refers to collection of neural tissue, including whole or parts of the brain, blood samples and blood fractions for future storage for use for transomic research. And this is useful because, as Rufus alluded to earlier on, it helps us to be able to gain a better understanding of the molecular mechanisms that underpins various neurological disorders, in particular stroke, but has benefit for other disorders such as Alzheimer's disease, Parkinson's, and so forth. But in Africa, neurobiobanking is still at the nascent stages, and that is why we did the ELSI study. And I want to share with you some 3 lessons in the interest of time that we've learned from the ELSI study. The ELSI study was mainly a qualitative study, and we had a multidisciplinary team. We had neurologists, people with expertise in genomics, neuropathology, public health, health promotion and education, biostatistics, communication law, bioethics, and sociology. So this was a very rich repertoire of individuals with varied backgrounds to interviews and surveys in Ghana and Nigeria. So the first lesson, lesson number one, had to do with, learnings on biological sample donation and informed consent. And here we sought to ascertain the awareness, the perspectives, and the predictors of willingness for biological sample donation, sharing, and informed consent preferences among our community members in Ghana and Nigeria. So we recruited over 1,000 participants with a mean age of about 39 years. And when we asked them about their willingness or their awareness and willingness to donate samples for neurobiobanking, what we found was that only up to about a third of our participants had heard about blood donation for the purposes of research purposes. And only about 25% were aware that you could use those blood samples for analysis in genomic research. And only 1/3 were also aware of the need to provide informed consent before your samples can be taken for genomic analysis. Well, we asked our participants, were they willing to donate their brain for research after their death? And interestingly, only 20% of them said that, well, I want to give my brain for research. And it was interesting because when we asked them, one of the things that they told us was that if they gave their brain over to research and then they appear before God after their death, and God asked them, where's your brain? What are they going to tell God? So that's one of the interesting reasons that came up from our discussions with them. But about willingness to donate blood for research, yes, there was a very high response rate that they were willing to donate blood, but not their brain for research. And then we also wanted to understand the sources of information on sample donation. And you realize that most of the sources of information for sample donation for research came from the hospital setting, close to 65%. So it means that radio, TV, seminars contributed very little to the patient's or participant's source of information for blood donation for stroke genomic research. The positive predictors for association or the associations for the likelihood of somebody willing to donate samples for biobanking and informed consent were being married, having a high level of education, tertiary level of education, being a student, and then certain ethnic groups were also associated with a willingness to donate blood samples and donate samples for biobanking. The learning number two, the key lesson number two, had to do with the legal aspects of biobanking. When we started the study, we were interested to know the position of the law on, stroke biobanking and genomic research. And we realized that this wasn't clear from our initial work that we did, looking at what law is available in Ghana or in Nigeria that regulates how people are supposed to donate their samples for research. So we reviewed the legal implications of biobanking, looking at the rights to dignity, the human right to privacy, the issue of consent and application of trust concept, and then the concept of medical negligence, intellectual properties. So all those were things that we looked at, and we couldn't find a lot of good guidance from the existing law. But what we found, which was helpful, was documents that were prepared by the H3 Africa Consortium on ethical guidelines on genomic research, given that sometimes you collect samples in Africa, it is shipped to either the US or the UK for these GWAS analysis. And the issue of transporting genomic or genetic data across the continent to other continents is also can be problematic. And so all these were nicely fleshed out by the H3 Africa. And we had some guidance on how to be able to share samples, share genomic data to promote research. The key lesson number three was also had to do with the issue of disclosure of genetic testing results. After you've collected a sample from a participant and said, look, I'm going to do GWAS testing, how do you feedback the results back to them? So we're interested in finding out the preferences, the perceptions, and the attitudes towards disclosure of genetic testing results. So we did a qualitative thematic analysis of key informant interviews of 61 participants from, we set up community advisory boards and then healthcare providers. And we found that there was a poor awareness of national guidelines on disclosure of genetic testing results. And so we're interested then in finding out what are the facilitators for disclosure as well as the inhibitors or the barriers to disclosure of genetic testing results. So the facilitators were having a good education. We found that if we had an enabling environment, now people have set up in Nkrumah where there's a genomic center for counseling. There's a center for counseling on genetic testing. So an enabling environment so that individuals can understand what we mean by a genetic test result. We also thought that if we had the religious and community leaders' involvement, it will help greatly because a lot of the information that has to do with genetics are shrouded in religion and beliefs. And so if we're able to get religious leaders to demystify genetic testing, the people we interviewed felt that it will help, as well as embarking on campaigns. And for instance, we've learned a lot from sickle cell studies that if you embark on campaigns, the acceptability of genetic testing results improves significantly. And then the big question then is, if you found that I have a genetic predisposition to stroke, if there's availability of therapeutic options, then it would enhance the uptake of genetic testing results. Of course, there are barriers, there are inhibitors to disclosure of genetic testing results, and most of them have to do with inadequate information. Fear of marital breakup also came out very strongly, that if I find that I have a loci or I have a SNP that increases my tendency for stroke and my husband is aware or my wife is aware and want to have kids, what do we do? There's also fear of stigmatization, fear of isolation, religious beliefs, healthcare worker attitudes, and lack of preparedness to accept results were all identified as potential inhibitors. So the conclusion from the neurobiobanking work that we've done so far, and we are preparing to enter the next phase, which is the intervention testing phase, is that culturally sensitive interventions for continuing education, increased awareness, and sustained engagement to equip all stakeholders in the genetic testing disclosure process is needed, particularly in our setting, so that most of the work that we seek to do in terms of, unraveling genetic markers for stroke, its uptake, its acceptance in terms of promoting precision medicine will be enhanced. So I would like to end here by acknowledging a ton of people who have been involved in the work. I'm happy to have done this with Rufus because he's a friend and a dear colleague. We've been working on this for almost 10 years now. But all these, Maiwa, Bruce, Albert, Dr. Albert Akpalu, ton of people have been involved in this kind of work, and it's not possible without their help. And we also want to appreciate all the sites who contributed data for this work, the NIH for their support, and thank you very much for your attention. I think I'll hand over to the organizers to find out if there are any questions.
Thank you so much to both of you for these presentations. We are happy to take questions. You can either post them in the chat or in the Q&A, or if you raise your hand, I'm also happy to grant permission to speak, which sounds like a funny thing to do. But maybe I can just get started. Prof. Sarfo, I was really interested in the fact that the people were really getting their information from the hospital rather than from friends or family or social media. And I guess that's very different from what we found in Zambia when we look at reasons why people decline lumbar puncture, for example. And I wonder if you think that there's if that narrative could be changed, if there was more education done in those spaces, that maybe this is just an unfamiliar concept, and so that's why it's not being discussed there. Or if you think that narrative needs to be changed, or we should keep it in the hospital where people are getting information. How do you view that side of things?
Yes, so thank you very much. Basically, I think the uptake of genetic testing results, I think it's been taken in silos. And when I say it's been taken in silos, there are certain diseases that, for instance, sickle cell, where without those tests, it's not possible to make a diagnosis. And so with that, there's been a big drive to educate the public on radio stations, on the TV. And because there's been funding for that, its uptake has been great. But when it comes to stroke, currently where we are in terms of genetic studies, I think we are more at the space of trying to understand the contribution of genetic loci to stroke occurrence, and then be able to now see whether those genetic loci translate to pharmacotherapy or novel treatments that could help improve or prevent stroke. So that's where we are now. And so it appears to depend on which kind of disease that you are sort of dealing with. And I think the overall concept of genetic testing will then have to be taken as a holistic concept that would inure overall to the benefit of our population, if we got to understand that this is what genetic testing would offer us in the long run, either for therapeutic, for preventive purposes. And I think that we have to look at the various age groups that we want to reach. If you wanted to reach a younger age group with genetic testing, you may need to use social media because they are big on social media. But if you wanted to reach the middle-aged elderly population, you may need to use a lot more TV, a lot more radio to be able to get through to them. So those are my initial thoughts on that particular issue. Rufus, do you want to add anything?
Well, I think you've covered that question.
And Prof. Akinyemi, and please, others post questions. I'm happy to keep asking questions, but even better if others do.
Right.
So we have one. So Dr. Chishimba had said, did education affect understanding of genetic studies is the first question. And the second one, I'm curious how you went about explaining genetic studies to someone with kind of moderate or minimal education. Particularly, how did you come up with terms and local dialects? And maybe we can start with that. And then I think the last one, how will this contribute to understanding of genetic studies overall?
Yes, so maybe, Rufus, do you want to start?
Oh, okay, you can start off, Fred.
Yeah. So I think the beautiful thing about the collaborative work we've done so far has been that when we started off, we had to translate a lot of medical terms into local dialects. And so if you wanted to explain the concept of genetics or heritability, you needed to use simple things like if your father is tall or if your father is dark and your mother is also dark, the two of them, when they give birth, you'll be dark. But if this is dark and this is fair, you'll be a mixture of those two. So it was really finding terms that people could identify and understand. And once we got those terms, those local dialects to be able to explain those, then we had to then apply it to, okay, so for a condition like stroke, for instance, it is possible that the genes in your mother and the genes in your father could contribute to your risk for developing a stroke. And so that is how we went about getting local terms to be able to explain what genetics is and then apply it to the context of stroke. And I think that we also were looking at, creating cartoons and things to be able to, use pictures to be able to explain concepts such as genetics to our participants. Rufus, do you want to join? But there was a second question. I think I didn't note that down.
Okay. Yeah, let me add to the first response on the first question. We actually, it is very true. We did translate many common genetic terminologies into local dialects in the study areas. And we actually convert all this into a diary, which is among one of the materials we published, especially from the African Neurobiology project. So that's an important product for that project. And this is because to ensure that, our interventions, which we have now developed and about rolling out, are context sensitive, essentially the language and the culture, especially for conditions that I was studying. And the ultimate goal of this is to use this as a model, to develop this something that will be useful for the rest of the continent. So that's the framework. Now, somebody did ask about education. I think the question was about level of education and willingness to subject to genetic test. Am I correct?
Yes, yes.
Right, okay. Yes, we found educational attainment to be a determinant of willingness to undergo genetic testing and also willingness to donate brains, even for research. In the Neurobiology studies, French put that a quarter of individuals. In another study in Nigeria, in Ibadan specifically, when we went across the West African solution, found it was to about 30%, also really among 400 participants in the geriatric clinic were willing to donate brains for research. Level of education was the most significant evidence factor associated with the willingness to donate for research. And the good news about this is that we need to just ensure everybody Costa Rica is educated. Everybody should go to school. We will ensure that you will not only enhance willingness to the research, but also people to community research, and then we also raise the resilience, protection against and we also found that one of our publications as well that individuals who were more educated were more likely to have lower body muscular risk factors. That came out one of the analysis we did and the data set. So being educated, having correct knowledge base is very important. I think also this has policy implications. African government, every education, they need to provide fair education. I mean, let everybody have a minimum level of education, maybe 6 years, 10 years, so that their health needs and knowledge, different conditions in the past. And that has overall protective benefits. Thank you.
Thanks. I think that's
People are raising their hands up. I think I saw Dr. Albert Akwalu. I'd raise their hand up. Dr. Akwalu, please go ahead.
Yes, thank you very much, Steve. Rufus. Hi. I was want to find out how far have you gone with IL-6. IL-6 was very exciting looking at an infectious hypothesis for stroke in Africans. But I think we haven't had any more luck with that loci.
Yes, the interleukin-6 variant was associated with ischemic stroke, the very first graded study. And the implication, what we thought was responsible for this, it could be a marker of higher level of oxidative stress amongst people of African ancestry. And this could be a result of exposures to infectious agents in the environment. I mean, malaria is all over the place, viruses and all that, they're all over the place. For instance, we know that this was one of the explanations given for why there was lower mortality among Africans during the COVID-19 pandemic. You know, that already has some baseline exposure to different, exposure. The other potential idea we had was that it also be a marker of exposure to other factors that could stress. Like air pollution, for instance. I mean, there are papers that are described as issue between PM 2.5. By and large, I thought it was a marker of something that they were exposed to in the environment. So that was the explanation for that. Of course, I know Fred has published some initial work on biomarkers, in stroke, JVP. So, looking at the genomics in our cohort, the metabolomics is the next phase, of what we're trying to get from for, so that all the storage. Yes, we don't do some piloproteomics work. There's a very nice marker that we found that is seems to be predictor of how the especially mortality.
Yeah.
Right up, and we tend to build on this to do larger population of larger sample for. So thank you for that question. And this may turn out to be an important either diagnostic or prognostic index. At least in agriculture.
Well, yes, and the other thing that the IL-6, which was a genetic marker, may just tell us that we are more at risk, or we have a proclivity towards accelerated inflammation. And this inflammatory process is what perhaps is bringing down our age of onset of stroke. If you look at the data, we have stroke 15 years younger than what happens in the West. So there is perhaps a proclivity towards accelerated inflammation among individuals of African ancestry. And it's also found in the Riga study, where IL-6 levels were significantly higher among Blacks than among Caucasians. So there's something in there that we need to continue to explore. And then on the comment of the education, yes, lower educational status means people are not likely to donate, but that also gives us an opportunity to know who to target when it comes to improving level of literacy on genetic testing, that those with lower levels of literacy may need more education to be able to increase their acceptability or their uptake of genetic testing. Over.
Great. I think we have two more questions. I know we're running out of time, but we'll try to get to them. The first one, I think, is a straightforward answer. Prof. Joey was wondering whether you came across known genetic stroke syndromes like carousel or CADASIL in the study.
For now, no. We haven't reported any.
Great. And then I think Dr. Gadama was wondering, so far the epidemiologic transition to non-communicable diseases and the burden of stroke has really been primarily attributed to multiple environmental and lifestyle contributions and changing epidemiology there. And so he's wondering, what percentage of this increased risk of stroke do you think is being explained by the SNPs that were identified from the GWAS study, or how do you put genetic contributions in the context of these environmental and lifestyle contributions?
Okay, yeah, thank you very much for that question. I would say that the data we're packing so far, Africa will not be sufficient to provide a very accurate answer to that question. However, we can just reference from the paper burden of disease results. One of the GBD papers, I think the 2016, 2019 paper very well, led by I've documented that when it comes to stroke risk factors, about 90% of stroke risk can be explained by risk factors. Academic factors, behavioral and environmental factors. So if you take that, that means there's a 10% risk that's either due to aging, which is the non-modifiable risk factor, or genetic factors. So that's what we know largely in terms of global epidemiology. However, there may be regional variations, the exact contribution of these different risk factors to our stroke risk. So these are the field in this exploration. Continue to explore these for the African continent specifically. But if the risk is 90% modifiable, then that means we need to do in terms of how to modify behaviors, improving our environment, and of course, controlling the metabolism of these factors.
And of course, so stroke risk is polygenic, isn't it? So on top of all these vascular risk factors that are associated with stroke also have their various genetic contributions. So it is likely that the genetics interact with environmental exposures, and that it may not be a clear-cut 80%, 20%, or 90%, 10%. But as we unravel more SNPs, we'll be able to then, estimate the contribution of these SNPs, which would mediate its effect via risk factors to stroke occurrence.
Right. One of the things we're also trying to do is to develop genetic risk scores. These have been found cutting edge globally. But one thing that is certain is that genetic risk scores that we develop for Asian populations, when they've been applied to African ancestry, they've not performed as efficiently as expected, which makes them into the aggregates from Africa to develop our own genetic risk scores, which will be more accurate, in determining the genetic information. So it's part of work in progress from design.
Thank you. I think that's a really interesting thought to end on here. This has been a really, really rich and informative talk today from both of you. So thank you both so much for giving the talk today. Thank you to everyone who attended today. We had a really great turnout. And I'm just really thankful that we were able to have this discussion. Please do check out our website, the worldneurology.com, where you can find out about future quarterly webinars as well. And again, thanks to everyone, and have a great morning, afternoon, evening, depending on where you are in the world. And Prof. Sarfo and Akinyemi, again, thank you so much for the talks today.